Hi class, in this recording we're going to be focusing on synaptic transmission, how we can take a signal, an action potential, from the presynaptic neuron and send it to the postsynaptic neuron. So let's dive into it. As we look at our synapses, some of them are going to be excitatory in nature in that they raise membrane voltage, others are inhibitory in nature in that they lower membrane voltage. And all depends on the kinds of receptors that are on that postsynaptic cell. So it's not so much the neurotransmitter as the receptor that's receiving the neurotransmitter. The same neurotransmitter can be either excitatory or inhibitory depending on the receptor that it binds to. Some of our receptors are going to be ligand gated, and others are going to act through secondary messengers. So as we're looking at these synapses, we're going to talk about three broad categories. We have excitatory, cholinergic, inhibitory GABA-ergic, and excitatory androgenic synapses. And these are going to focus on the three of those broad categories of neurotransmitters that we talked about, the acetylcholine, and then we're going to have the monoamine, and then we're going to have the amine-based neurotransmitters. So let's dive into it. As we look at our cholinergic synapses, they are based off of acetylcholine. The common abbreviation for acetylcholine is capital A, capital C, lowercase h, ACH. So, I could spend a lot of time reading this out loud to you. Let's see if I have a fig. Here we go. I'm gonna camp on this slide. So you can read the previous slide if you want to. I like talking through pictures more than reading text out loud. So we're going to have the action potential. Let's shift to bright green here and clear the screen. So we're going to have our action potential reach the end of the axon terminal. When it reaches the end of the axon terminal, calcium voltage-gated channels open up and calcium ions rush into the cell. Those calcium ions, I'm going to shift to red, are going to bind to the synaptic vesicles. When calcium binds to that synaptic vesicle, the synaptic vesicle will fuse with the plasma membrane and exositize the acetylcholine that's being stored in its synaptic vesicle. We're then going to have the empty synaptic vesicle go back into the cell and get repopulated with acetylcholine. After that acetylcholine was released, though, that acetylcholine is going to go into this space in between the two cells. And we refer to that space in between the two cells as the synaptic cleft. As acetylcholine gets into the synaptic cleft, acetylcholine will bind to a ligand receptor. And this is a sodium voltage, this is a ligand sodium ion channel. So when two acetylcholines bind to this ligand or this ion channel, it opens up and the sodium ions that are extracellular will rush into this postsynaptic cell. And as we add more and more sodium ions to the inside of the cell, we're going to change the voltage on the inside of the cell, and then we'll have potassium voltage-gated channels open up. Those potassium voltage-gated channels will then allow positive potassium ions to rush out of the cell, and we're going to have the movement of our ions now, and we're going to initiate a change in voltage that initiates an action potential that then initiates a change in charge that's propagated down the length of the cell. So we have our presynaptic cell, our postsynaptic cell, and to have an electric signal or action potential go from the presynaptic cell and make it to the postsynaptic cell, we need to bridge this synaptic cleft, this space, using neurotransmitters. Now, let's back up. I want to make sure I don't get too far ahead of myself here. As we're looking at GABA-ergic synapses, these GABA-ergic synapses will employ gamma-aminobutyric acid, hence GABA, as their neurotransmitters. So we'll release GABA into the synaptic cleft, and those GABA receptors are going to be chloride channels. Acetylcholine receptors are sodium channels as opposed to chloride channels. And as the chloride channels open up, instead of having sodium ions rush into the cell, we'll have negative chloride ions rush into the cell, making the inside more negative and lowering the voltage. And because we lower the voltage, we are inhibiting the neuron from initiating an action potential. So this is a key idea here. We talked about with hyperpolarization 2 recordings ago. If you lower the voltage of the membrane, you inhibit an action potential. Rising or raising the voltage of the membrane will promote or excite the action potential. I got a little far ahead of I got behind. I forgot to mention these steps right here. So as I'm looking at these steps right here, we need to ask ourselves, what happens to the acetylcholine after the acetylcholine has been dumped to the synapse? If the acetylcholine stays in the synapse forever, the acetylcholine is going to keep binding to the sodium ion channels and continually initiating more and more and more action potentials. That's not necessarily a good thing. We need a way to stop initiating the action potentials. And you can see that right here. ACHE stands for acetylcholine esterase. And what ends up happening is we have digestive enzymes in the synaptic cleft themselves, this acetylcholine esterase. Acetylcholine esterase will bind to those little green acetylcholine molecules and chop them in half. The acetate, which is part of acetic acid, will then diffuse out to the rest of the body. The choline is going to be reabsorbed into the presynaptic neuron, and that choline is recycled to make more acetylcholine, which is then put back into synaptic vesicles so we can repeat the entire process over again when we have the next action potential arrive. Okay, let's focus on our androgenic synapses. When we think of androgenic synapses, I want you to think of them as primarily excitatory. So when we look at these androgenic synapses, they're going to rely on norepinephrine frequently. Occasionally epinephrine. NE is also known as noradrenaline. So when we think of norepinephrine versus noradrenaline, generally speaking, this may be an oversimplification, I think of the European versus the American terminology. So as we're looking at this norepinephrine, it's a monoamine based neurotransmitter, and it's like neuropeptides, they act through secondary messenger systems. We talked about secondary messenger systems with chapter 3 of this class when we talked about G-coupled protein cascades. The 2 most common secondary messengers are calcium ions and cyclic AMP, oftentimes abbreviated as CAMP. And in case you forgot, AMP stands for adenosine monophosphate. So it's instead of an ATP with 3 phosphates, it's an AMP with 1 phosphate. Now, these receptors are not going to be ion gates, but instead it's a transmembrane system. It's a G-coupled protein cascade. Because there are more proteins involved and more steps involved, these are slower to respond than our cholinergic or GABAergic synapses. However, there's a distinct advantage. Because it involves that G-coupled protein cascade, we can have enzyme amplification. One single molecule of norepinephrine can produce a very, very strong downstream effect or very, very strong impact because there's the amplification of the signal with the secondary messengers. So as we're looking at this androgenic synapse, we're going to have our receptor, and this neurotransmitter receptor can have norepinephrine bind to it, represented by that little green molecule. When norepinephrine binds, it releases the G protein. The G protein will then go to adenylyl cyclase. Adenylyl cyclase turns ATP into cyclic AMP. Cyclic AMP can do lots and lots of different things. There's multiple things it can do. It can turn on enzymes. It can activate ligand-gated ion channels, or it can turn on genes. It can initiate genetic transcription. If it does that, genetic transcription is going to make more enzymes. So either it can turn the enzymes on directly, it can make us make more enzymes. Regardless, we're going to have a metabolic change, a change in cellular chemistry. So these androgenic synapses are slower, but they have very profound effects when we activate them. Now, how do we turn signals off? We don't want our synapses to be running constantly. We need to stop them. We need some way to stop releasing the neurotransmitter. Now, if we have our action potential on the presynaptic cell stop, that presynaptic cell will, well, stop releasing neurotransmitters. Neurotransmitters are only going to linger in the synaptic cleft for about a millisecond, and then they're typically cleared from that synaptic cleft. So, I talked about it briefly with a picture several slides ago. Let's talk about it in more detail right now. Step 6, 7, 8, processing that acetylcholine. So the acetylcholine right here, let's go back to this stage right here. That acetylcholine would be broken down by an enzyme called acetylcholinesterase. Acetylcholinesterase is then going to send the choline to receptors and the presynaptic neuron so that the choline can be reabsorbed and recycled and turned into more ACH. We're also going to find that the axon terminal can reabsorb the neurotransmitter by endocytosis. So monoamine transmitters can be broken down after reabsorption by monoamine oxidase. And we'll talk about monoamine oxidase in more detail when we get to some of the pharmacology. These neurotransmitters, in step number 8, these neurotransmitters can also diffuse to the extracellular fluid. So as we're looking at neurotransmitters, some of the neurotransmitters will be broken down by acetylcholine esterase. Other times the neurotransmitters will just diffuse away. They don't have to physically stay here. There's nothing containing them within the synapse specifically. So to review, how do we get rid of the neurotransmitters from the synapse? We can have an enzyme like acetylcholinesterase destroy them. We can have the axon terminal reabsorb them directly, or they could diffuse away into the extracellular fluid. What's a neuromodulator? Neuromodulators are chemicals that have long-term effects on groups of neurons as opposed to little short effects. So what can a neuromodulator do? A neuromodulator can alter the rate of neurotransmitter synthesis, release, reuptake, breakdown, or they can adjust sensitivity of the postsynaptic membrane. So neuromodulators have profound longer-term effects. Nitric oxide, NO, is a very simple neuromodulator. We talk about it frequently. It activates secondary messenger pathways, famously relaxing smooth muscles in our bodies. Those neuropeptides that we talked about in our last recording can be chains of amino acids, typically between 2 and 40 amino acids, that will act as a neuromodulator. When we think of enkephalins and endorphins, these are two categories of neuromodulators or neuropeptides that act as neuromodulators that inhibit pain signals in our central nervous system. You've probably experienced something like this before when you've been running around outside. Maybe it's an athletic event, maybe you're just having a lot of fun in nature or hiking, and you get a scratch, a bump, or a bruise, and you don't feel it until the next day, or at least until the activity is over. And that's because of those enkephalins and endorphins that have been released. That's all we have for our discussion on neurotransmitters and our synapses. If you have any questions, please feel free to post them on the class discussion board, shoot me an email, and as always, happy studies.